The Drosophila heart is a simple tubular structure with a central lumen that pumps hemolymph around the fly's body. Rolf Bodmer and colleagues at the Sanford-Burnham Medical Research Institute in La Jolla, California, are interested in Drosophila heart morphogenesis, a process that begins with the alignment of cardioblast cells at the dorsal midline of developing embryos.
So they have these apical protrusions that come together, and basically the cardioblasts then curl around to form a lumen between the contralateral cardioblasts.
Lumen formation is promoted by the extracellular matrix signaling protein SLIT, which is thought to inhibit intercellular adhesion in the organ's luminal domain. Bodmer and colleagues, however, were interested in the potential role of Cdc42, a small GTPase already known to regulate adult heart function in flies and mice. Postdoc Vogler therefore examined the developing hearts of fly embryos lacking Cdc42, and found that the cardioblasts often failed to align and contact each other at the dorsal midline.
But once they make the contact, you don't really get this luminal space there. So instead of having these nicely curved, bent cardioblasts, they are more flattened.
SLIT still localized between contralateral cardioblasts, but the cells failed to form a distinct luminal space. This effect was even more pronounced in cardioblasts overexpressing dominant negative Cdc42. In contrast, overexpressing constitutively active Cdc42 induced the formation of ectopic lumina between ipsilateral cardioblasts neighboring each other on the same side of the dorsal midline. Vogler et al. Then found that the heart defects in Cdc42 mutant embryos were exacerbated by deleting one copy of ZIPper, the gene encoding the motor protein non-muscle myosin 2. This suggested that ZIPper and Cdc42 work together to control heart morphogenesis. Indeed, embryos completely lacking ZIPper showed similar defects in both cardioblast alignment and lumen formation.
So we then obtained from Dan Kierhart's lab the GFP tag, non-muscle myosin, that we then overexpressed in wild-type hearts. So when we took this movie, that was like one of those, eureka moments. You see like this beautiful dynamic wave pattern of zipper, accumulating at the apical site of that cardioblast, which, hasn't been reported before. These pulses were like something very, very new to us.
These apical myosin pulses might induce contractions that help draw cardioblasts together at the dorsal midline. After dorsal closure and cardioblast alignment, zipper relocalized to the nascent luminal domain, supporting its involvement, along with Cdc42, in either lumen formation or maintenance.
We then started to manipulate Cdc42 and then saw effects on non-muscle myosin, basically loss of this repetitive pattern when you inhibit Cdc42 and this sustained accumulation when you activate it.
Non-muscle myosin is typically regulated by the Rho GTPase, but inhibiting Rho1 or its downstream effectors had no effect on heart morphogenesis. Cdc42 could control non-muscle myosin by regulating the assembly of actin filaments that then recruit zipper to specific regions of the cardioblasts to direct the cell's alignment and subsequent shape changes. Vogler et al. Therefore examined whether actin-nucleating formin proteins worked downstream of Cdc42 to regulate zipper and heart morphogenesis.
The formins themselves, we didn't see any phenotypes when we knocked them out, but the Cdc42 double mutants have like a very severe phenotype that's like even worse than what we see with Cdc42, showing that Cdc42 and formin activity are controlling the formation of the heart formin.
On the other hand, overexpressing constitutively active formin proteins, even in the absence of Cdc42 activity, induce the formation of ectopic lumina surrounded by non-muscle myosin and luminal markers such as SLIT.
We get a complete de novo formation of a heart lumen with all the factors that we know of at an ectopic position. I would say that's really new, that formins not just control like a little event, but that if you manipulate formins, then you can relocate like an entire structure to a different place.
CDC42 and formins therefore combine to regulate the dynamics of actin-myosin filaments that control the shape of Drosophila cardioblasts as they assemble into a tubular heart structure.
Before closure, there's like a temporal accumulation of non-muscle myosin at the apical side of these cardioblasts that is dependent on CDC42. And if you look at a movie of a closing heart, you can see that these cardioblasts, each of them have some kind of apical constriction, as if they are all individually pulling. So a role or function of this temporal accumulation of non-muscle myosin at this stage could simply be that it supports the closure of the heart. After closure, what we see is that as the lumen is being formed, we get, again, strong pulses of non-muscle myosin at the luminal domain. We think that this is something that either keeps maintaining the luminal space or enhances the luminal space. We don't know which of these two it is, but it could just be that it basically have actin-myosin opening the luminal space and then having slits kind of as an anti-adhering factor that then keeps this open.
The CDC42 formin zipper pathway seems to function independently of slit. For example, zipper's localization is unaffected in mutant embryos lacking the slit receptor. The researchers are therefore interested in determining what lies upstream of CDC42 during heart morphogenesis.
So what is giving the pattern? What is the instructive information that is then executed by these more downstream CDC42 and actin-myosin? So those are more the puppets that actually are doing it. And so we are interested in trying to figure out, what are the critical puppeteers that are directing what's happening where to form this orchestrated cardiac morphogenesis.
In the meantime, however, you can learn more about how CDC42 and formin activity controls non-muscle myosin dynamics during Drosophila heart morphogenesis in the paper by Vogler et al., published in the September 29, 2014 issue of the Journal of Cell Biology.